When I teach genetics, we go to lab and we start talking about fruit flies, and students start counting fruit flies and do experiments with fruit flies. And I think an obvious question that a student could ask is, why do we care about fruit flies? And the honest answer at some level has to be, well, we don't. And I say that as someone who spent 4 years of their life in grad school studying fruit flies. We don't really care about fruit flies at all right, because there's more than 1 million organisms on the planet which we've identified as species. There's far more that we haven't identified. And so why would you pick one? Is one any more important than others? Then the answer is no. So why do we care about fruit flies? Well, a couple of answers to that. One is we care about genes. In genetics, we want to study genes. And fruit flies are packages of genes. So in a sense, I don't really care about the fruit fly itself, but I do care about the genes that it carries, because this fruit fly then allows me to study genes and how they work. Now, and there are just some advantages. Fruit flies are small. You don't need a whole lot of space to keep them. They're cheap. They have a short generation time. If you're looking at how genes are passed down from one generation to the next, if you were to make this female fly, then you could have her offspring within, say, 11 to 14 days, and then the grandchildren, the F2 generation, 2 weeks after that. So in a month, you could study 2 new generations of flies. If you were doing genetics experiments on like elephants or horses, I mean, it would take you the rest of your life to do that. And so this just happens to be a great way of studying genes. Now, why would you want to study fruit fly genes? Well, that's a great question. And I'd spend a lot of time answering that, but this is meant to kind of be an introductory video. So let this be then the short answer. The long answer to that is because humans have so many homologs of Drosophila genes. Humans have genes. We have genes which, pump ions across cell membranes. We have genes which transport things. We have genes which direct cell division. We have genes which detect light. We have genes which allow ions to pass or bind hormones, etc. Well, a question could be asked, are these genes unique to humans? Do other organisms have them? And the answer is no. These genes are almost never unique to humans. Other organisms do have them, such as flies. All right, and then another question might be, well, who cares? Well, why do we care that flies have these genes? Well, one big answer is, if you want to identify a gene and study a gene, it's extremely difficult to begin your study in a complex organism like ourselves. It is far easier to start with a simple organism and then identify the gene, find out its sequence, and then take what you've learned and apply it to a more complex organism. So, for example, if you were to discover, say, a gene expressed in Drosophila development in the larvae, you could study it, you could get its sequence, you could find out where in cells it was expressed, etc. Well, once you had a sequence, you could then make a probe based on that sequence. You could get a molecule that sticks to this protein, whatever it is. Having done that, you could then take vertebrate embryos, like a chicken or a frog embryo or a pig embryo, and say, all right, well, now that I have a probe that sticks to this gene that I've just discovered and its protein or its protein, is there a version of this gene expressed in the embryos of vertebrates? And very often there is. And then you ask, well, what does this gene do? And then once you study invertebrates, you can then ask, well, do humans have a version of that gene? Very often we do. And very often that's how new human genes are discovered. You wouldn't have discovered the new human gene in we, the complex organism, if you hadn't found it first in a simpler organism. And you might say, I don't know if I buy that. I mean, fly eyes, what do they have in common with human eyes? Virtually nothing. What would fly wings have in common with human, structure? Very little. Well, it turns out what determines whether flies have the normal brick red eyes or scarlet eyes or brown or white. It's a protein. It's a channel which pumps pigment. It's what's called an ABC cassette transporter. And so when you're studying scarlet-eyed flies or brown-eyed flies or white-eyed flies, you're looking at mutations or mutants in this specific gene. Well, once you know what that type of transporter is, what its sequence is, where it is on the cells, how it interacts, do humans have versions of the gene? It turns out we do. Now that's like making our eyes scarlet. Some of our genes are pumping cholesterol, determining our risk to heart disease. We're pumping toxins out, or elements of chemotherapy out of cells which cancer cells might use, which would then determine the efficacy of chemotherapy treatments. There's a whole set of these transporters in humans, maybe not doing the exact same thing, but now that we know what to look for, we can identify them in humans. So what causes sepia eyes in flies is a glutathione S transferase. Is that what's determining brown eyes in humans? No. But GST enzymes are very important in humans in determining asthma susceptibility. And we could go into others. Do we, have bar eyes? Or do we have mutations like some flies can where the legs come out the head? It's known as antennapedia. The answer is no. But those were mutations in DNA binding proteins called homeodomains. Turns out we have hundreds of them, that they're critical, especially during embryonic development. And we wouldn't know much about them, if anything, if we didn't first identify them in a simple organism and say, now that I know what this protein looks like, I know what to look for. Do humans have a version of this? So once again, geneticists very often study fruit flies, or E. coli, or a nematode worm, or mice, or some experimental organism, and an outsider could legitimately ask, really, why do you care? And the answer is, well, we care about genes. We want to understand genes. And this organism is just the package that holds the genes that allows us to study them. Why would you study flies? Well, here's a fly stock center where you can order stocks. And their homepage starts off with this statement. It's estimated that 65% of human disease-causing genes have a functional homologue in Drosophila. So in other words, if someone's kind of implying, why are you studying flies? Why wouldn't you study humans instead? Well, very often it's the study of flies or some other research organism, which then gives us a better understanding of, a much more complex individual like humans. So we don't have to study flies. We could study zebrafish or nematodes like C. elegans, or E. coli, or yeast, etc. But if you were going to study flies, if this is your research organism, for some parts of lab, you have to understand it. So let's just kind of go through some basics. Once again, you're not going to do very good research if you don't really understand your research organism. So flies are obviously insects. As such, as adults, they have three body parts: a head, a thorax, and an abdomen. The head has the big compound eyes, the antenna. The thorax has three pairs of legs and one pair of wings. Most insects have two pairs of wings, but flies, what makes the order Diptera distinctive, among other things, is that one of the pairs of wings has been modified to make a balancing structure known as a haltere. So, there are the antennae. One can distinguish between males and females. The males are a little smaller. Also, the males have a darkly pigmented abdomen. You can see that black tip there, that's absent in females. Also, there's little bristles on the forelegs known as sex combs, which are only present in males. So one can distinguish between males and females. And if you were setting up experiments where you were, mating flies or looking for sex linkage and asking, is this trait more likely to affect males than females? Well, you have to know the difference. I've had sections where, there's a month before the data becomes available, and then we count all the flies and we put all the notes on the board. We're going to write a lab report, but one of the students couldn't tell the difference between males and females, and thus the data makes no sense, and it really affects the entire class, because we weren't expecting that. Well, that's not actually representative of the results. But obviously, if you can't tell males from females, studying sex linkage then becomes difficult. Just a little bit then on the fly life cycle, where males court the female. So no mating occurs until the female approves of the male. There's a female here. And so then the male approaches the female, then begins to tap her legs. During this and the subsequent processes, both can be exposed to their pheromones. They produce chemicals in their cuticles, which are signaling molecules. Among other things, this could signal the species that they are in. So for example, there's some fruit fly species that look very much like each other, but you would want to distinguish because, flies of this species don't mate with flies of that species, or at least they don't produce fertile offspring. So here you can see, once again, the female doesn't have the darkly pigmented abdomen nor the sex combs, while the male would. So the bare minimum that the male must have, and this is important for what I'm about to say, is pigment around its genital arch. Because you might say, oh, that's pretty easy to identify, that dark pigmented abdomen. And that's true, but it's perhaps not quite as dark in very young males. So it's easy enough to tell the difference between males and females, especially if the male's a little older. You can see the pigment on the male abdomen and then those sex combs. It is a little bit harder when the male is young. I'm sorry. I thought I had an image there as well. But then you can still see a genital arch, but you don't see as much pigment. And so then, I'm sorry, as I was saying, here's a male courting the female. He's reaching out and touching her, and then he begins to wave his wing and vibrate it to make a species-specific song. Once again, there are multiple species of fruit flies, and this female wants to know that this male is the appropriate species. He extends his proboscis to her genitalia, and then later will begin to curl his abdomen and attempt to mate. But once again, this is something that the female will accept or no. And there are some things which make her less likely to accept. So for example, if she is already mated and is laying eggs, then she will typically not mate until she has exhausted the sperm. So many animals, including flies, can store sperm after mating. And so a female may not choose to mate again until her sperm has been, the stores have been used up. So flies lay small oblong eggs that have two little extensions known as floaters, because very often they're laying it in a semi-solid medium, and this prevents the eggs from sinking. When fly eggs hatch, they do not hatch as small flies, but rather as these worm-like larvae. That is true of many insects. Butterfly young don't hatch as butterflies, but as caterpillars. Beetle larvae do not hatch as beetles, but rather as larvae. And then the same would apply to flies. The fly larvae are eating machines, so they're just going to be in the medium, just feeding and feeding, and go through several what are known as instars as they grow and ingest and take in food. And then when they reach a specific size, they will crawl out of the medium and then form a pupal case, just like a cocoon that a caterpillar would form. In this pupal case, they then reorganize their tissues so that they change their body shape from that original larval state to the state of the adults, and then they eclose from the pupal case then as the young fly. Now they're very often a little lighter in color for a bit once they eclose. So here are just a few stills of some of what I had said. There's that balancing organ. This is obviously a female. When females are under anesthesia, they often then begin to lay their eggs, and their eggs are laid very often in a soft medium. And then the larvae then travel throughout the medium feeding before they crawl to the sides to form a pupal case. So these are larvae. Here's one that has formed a pupal case, and it's in the pupal case that then they reorganize their tissues to the adult form. If you have a vial of flies, you might then see empty pupal cases here after the adults have already eclosed. So once again, there's a pupal case with a fly developing in it. The one that's more transparent has already eclosed. Once again, there's that holt here. And as I will get to in a second, we'll talk about the normal phenotype of flies. Notice that and the normal condition is that they have only one pair of wings and that the wings extend past the abdomen. They have 3 pairs of legs, and males can have holters on their forelegs. That males then have pigment around the genital arch that the females do not have. So you can distinguish between male and female. Although one has to be careful with very young flies. So very young flies are often lighter in pigment. They often have this dark spot where they are still processing, some metabolites from the pupal case. And one has to be careful, for a couple of hours, because these are males, even though they don't have their tip of the abdomen as darkly pigmented. But nevertheless, you can then notice there is dark pigment around the genital arch that females don't have. So even in young flies, which have just left the pupal case, you could still distinguish between males and females. Their wings are not unfolded when they eclose, and they might look like this. Very often students feel that they've discovered a new mutant fly. This isn't a mutant. Those are normal wings. They just have not yet eclosed. The video that you're about to see will go through just some of the variations that we study in mutants, because very often now if you're studying them, you're going to distinguish between the normal condition, which, for example, with eyes is a brick red color. That's what we call the wild type. And then there are variant mutant colors, which can then be different. Eye size can be affected. Eye shape can be affected. This is what's known as bar eyes. You can have wings be absent or reduced, all right, or wrinkled, or shorter than usual. Mutations can affect body color. The normal wild type color is a tan body color. Some mutations cause darker body colors, and then some mutations cause lighter body colors. Once again, it is the tan, which is the wild type. Now, when I say wild type, I obviously have to stress, I'm talking about Drosophila melanogaster, this species of flies. There are other species of flies, and their normal condition, their wild type, then varies. And so, as we're looking to study genes, we then often find them by finding abnormal flies that must have a change in a gene. And then we ask, what gene has been changed? Now, once again, you might say, who cares about a gene that would wrinkle wings? Well, it turns out humans have a homologue of that. Not obviously for wing shape, but it actually has a role in cancer. The apterous gene, we obviously don't have flies, but we have homologues used in embryonic development as a signaling factor. So once again, even if you didn't think that you could directly relate, the study of, say, wings on a fly to humans, given that we don't have wings, no, the genes which are used to signal, say, put a wing here, or have the cells of the wing do this process, those could be genes which humans have, and obviously not using for wings, but using, to have other signals be sent, or do other cellular processes. So once again, wings can vary, as can eye color. The normal eyes are a brick red, but some eyes are more of a scarlet, some eyes are more of a brown, some eyes are white. Body color can once again change. So there's one mutation known as ebony, another known as black. There was one mutation known as yellow. Okay, so not to get too into flies, obviously there have been hundreds of thousands of papers written on flies and their genetics. Some of it is fly specific, but most of it is now, once we understand this mutation that has been discovered in flies, then we could now see if we can apply that information to other organisms like ourselves. Sex linkage, for example, was first discovered with white-eyed fruit flies. And then once that form of inheritance was understood, the question was asked, are there any traits in humans that follow that pattern? And the answer turned out to be yes, like hemophilia, color blindness, and muscular dystrophy. And so just very quickly to introduce flies that you might use in a genetics lab. Do we care? Well, to some degree, no. Once again, we're studying genes. They just happen to be in flies. If it was convenient to study them in yeast, we would study yeast. We would study nematodes. We just want to study the genes. But once we've decided that we're going to study flies as, say, a research organism, we certainly need to know a little bit about them, or else our experiments, we're not going to design them well. So that's what this video was meant to introduce.